Thank you very much. If you'd all please take your seats. I think we can get started. It's so exciting to see everybody here today. We're already off to a great start at the meeting with the announcement of the first set of awards. I want to thank everyone for being here. Welcome you on behalf of my co-hosts and myself, Ken Frazier, whom you saw earlier, Mike Rosenblatt as well, and I'm Marc Tessier-Lavigne. I'm the co-founder and CEO of a company, Xaira Therapeutics, that focuses on applying artificial intelligence to transform how we discover and develop drugs. I'm also still a Stanford affiliate. I'm not, what it says on your program, also the CEO of NOETIK. I do not have that third title. The CEO of NOETIK is here. So I just have the first two affiliations. But it's great to be here. I know that AI is everywhere right now. It's in the news every day. We are all using it. Everybody's using ChatGPT and other tools. And it's certainly an AI moment. There are many markers of this. I think for many scientists, one of the key inflection points was when AlphaFold 2 was released. That was just 4 years ago. Hard to believe it was only 4 years ago. I think for the rest of the world, the AI moment was, of course, the release of ChatGPT in November of 2022, just 3 years ago also. AI has been on a tear. It was on a tear before that, but it's been on an inflection since. And just as one marker, 2 days ago, NVIDIA, which of course makes the GPUs that power it's a leader making the GPUs that power the AI revolution, just passed $5 trillion in market capitalization. Think of that. One company, $5 trillion in market cap, not billion, trillion. And that just shows the incredible demand that there is for their products, which of course is powering the revolution. AI, of course, is fueled by three things. You need algorithms, models. A key transformation there was the 2017 publication of the transformer model, which of course enabled large language models like ChatGPT. So you need algorithms. That's not the only one. We'll talk about some of those here today. You need compute. That's NVIDIA and GPUs. And then you need data, massive amounts of data, also something we're going to talk about. Those are the three legs of the stool for AI. I think it's fair to say that everybody is focused on what the impact of AI will be on human health and in particular on drug discovery and development. If you listen to the leaders of some of the leading companies, whether it's Jensen Huang at NVIDIA, Sam Altman at OpenAI, Dario Amedi at Anthropic, when they're asked what is the top application, they all respond with the same thing: human health. Human health. And the purpose of this panel is to discuss the promise of AI, but also the reality of it. I think it's fair to say that there are a lot of AI optimists, enthusiasts. I count myself among them. There are also AI skeptics. Perhaps there are some in the room here today. And I think it's important for us to take stock of where we're at with the technology, reflect on it, and see how we can accelerate the benefits and also overcome obstacles in the way of realizing those benefits, but also think about the downside. So that's what the panel seeks to address. In thinking about this problem in the context of drug discovery and development, which will be our focus, it's useful, in my view, to look at it through two lenses. The first is the trajectory of drug discovery and development. There are three things you have to do to make a drug. You first have to choose a molecular target. What is it that you're going to make a drug against? Typically a protein, but sometimes other entities. Then you have to make the drug to have the intended effect in the intended tissue, typically to switch off that protein, or sometimes to switch it on or otherwise modify it. And then you can have the best drug against the best target. You need to know who are the patients who are most likely to respond, because if you go into clinical trials and put in the wrong patients, the drug will fail. We know that 9 out of 10 drugs fail in clinical trials. I think everybody knows that statistic hasn't budged in 20 years. And we know that the major cause of failure is the failure to find the right patients for the target and the drug. So, and that's why we have this massive attrition, 9 out of 10 in the clinic. That doesn't count all the failures up to that point. Extended timelines and $2 to $4 billion to go from target to approved drug, factoring in the failures. So we have to do better. And the question is, what can AI contribute to improving the success rates, shortening the timelines, reducing the costs? and also what are the implications of the application of AI in this context. So for that, and before I go to the panel, just I do want to seed a little bit thinking about promise and reality. In my view, there are three levels at which AI is operating. One is at the level of operation and logistics and accelerating clinical trials. And there, I think the reality is that AI is already making a massive difference through the kinds of algorithms and approaches that we have. So that's something that's already galloping, if you will. In the middle, there's making drugs. And here, AI in particular is helping with molecular design, designing molecules. And that's already off to a great start, not least because of advances like AlphaFold and then David Baker's work on RF diffusion, RF antibody, just 2 years old. Again, it's all happening very quickly. And we're going to discuss that also, so the contribution to molecular design. And then one of the biggest differences to the success of our enterprise is if we can do a better job of assessing targets and matching them to patients, this problem of patient stratification. And there, I think the AI is making a difference, but it's perhaps not on the same time scale as that. So we have an all-star panel here to address those three layers. To my left here is Tom Miller, the co-founder and CEO of Iambic. He's going to tell us about Iambic and about molecular design, that particular step of the application of AI. Next to him are Ron Alfa, the CEO of NOETIK. He'll tell us about NOETIK, and Marinka Zitnik, who's on the faculty at Harvard in the Department of Biomedical Informatics. They're going to tell us about this problem of target identification, matching targets to patients, and the progress and promise there. To their left is Michelle Longmire, who is the co-founder and CEO of Medable. And she is focused squarely on this problem of accelerating clinical trials by focusing on applying AI to operations and logistics. And then to the left, to expand our thinking, we're very fortunate to have the Reverend Father Andrea Ciucci, who is from the COO of the Pontifical Academy for Life in Vatican City. He's the General Secretary of the Renaissance Foundation. His job, he is assigned within the Vatican to think about this AI moment, what the implications of AI are for society in general. And this is all fields, not just human health. But today we're fortunate to have him here to reflect on the implications of AI for human health. And his role in some ways, whereas the others are going to be answering questions, his role will be to make sure that we ask the right questions about the application of AI. Without further ado, I'd like to get started, and I'm going to ask each of the panelists to briefly introduce themselves and tell them first about their own specific focus in their company or in their lab, in their endeavor, and the advances, what their vision is for that, and also what advances they've made, just to place each of them. And then we're going to broaden the conversation after we've done that to talk about the promise and the obstacles in each of these fields. So Tom.
Yeah, thanks so much, Mark. And it's a wonderful opportunity to be part of this panel and to speak to this audience. I'm Tom Miller. I'm one of the co-founders and CEO of Iambic Therapeutics. I was previously a professor at Caltech for over a decade, working at the interface of computation and chemistry. And really, we became inspired and motivated by the opportunity to use new AI-driven technologies to really try to accelerate the delivery of better medicines to patients. And Iambic is really built on that thesis. At Iambic, what we do is we primarily focus on small molecule drug discovery. It is a flexible platform. We take vast amounts of different sorts of data, large repositories of heterogeneous data. We train large foundation models on the basis of that to allow us to holistically design compounds that will be either best in class or first in class, depending on the application. We combine that with high throughput experimentation. So you don't want just to do the AI design, but you want to immediately make those molecules, you want to test those molecules, you want to actively learn on the basis of that data. We do that for thousands of molecules on a 7 to 10 day loop. So being able to ask and answer questions very quickly. The proof of the pudding is in the eating, and the ultimate measure of any platform is what comes out and what actually gets delivered to clinics and to patients. We're excited about how that is working at Iambic. We have 3 programs that we've announced. The first of those has already reached clinic, and 2 others that are coming shortly. Our most advanced program went from program launch to clinical entry in less than 2 years. This is a brain-penetrant HER2 small molecule inhibitor with over 1,000-fold selectivity versus the dose-limiting off-target tox associated with EGFR. We're very excited that that program has not only entered clinic so quickly, but it is proceeding very quickly through clinic as well. Just last week at ESMO, we were able to report the data, the preliminary data from the phase 1b trial of that, completing dose escalation, and demonstrating both broad activity and a differentiated safety profile of that. So we're excited to see AI not only hold promise, but also execution, and very, very happy to discuss further with the panel on those matters.
Great, thank you. Ron.
Yeah, thanks, Mark. Again, yeah, great to be here. So Ron Alfa, co-founder, CEO of NOETIK. Prior to NOETIK, was an early employee and head of research at Recursion Pharma, where I was for 6 years, essentially from seed stage through IPO. And largely my motivation for founding NOETIK was this core problem of translation. So how do we know which drugs are going to work in which patients? And I think Mark set it up very nicely that we haven't really impacted clinical success in decades. That is still a huge failure in many areas, but especially in oncology where we're very interested. And so really the core vision behind the company is, can we build foundation models of patient biology that can help us understand which molecules are going to work in which patient populations. So essentially matching therapeutic biology to patient subtypes in a way that's more complex than say individual genomic biomarkers. And so when we founded the company, we had this crazy idea. Let's go. This data doesn't exist. We're just going to have to generate it. And so, since the beginning of the company 3 years ago, we've been sourcing human tumor specimens and processing them into really a rich multimodal data set that covers pathology, protein, and then whole transcriptome spatial. And so we're essentially developing this paired data set that we can use to train transformers in a self-supervised way. So what that means is we're not giving the models any labels. We're asking the models to essentially learn biology from this very complex, multi-layer data set. And so this was a really ambitious goal, and actually it works. I mean, it's phenomenal that if you generate a really powerful data set and you scale that and spend a lot of money on GPUs, these models can actually learn biology. So there are 2 examples that I'll reference that really kind of shocked us and helped us sort of see that this is, we're moving in the right direction. So first, we can take hundreds of lung cancer patient samples. We could generate this data, and we could train models to identify different patient subtypes. And it turns out that those models will learn which patients respond to immune checkpoint inhibitors without the labels. So the models will actually do better than, let's say, PD-L1 as a protein label to understand which patients are going to respond in the clinic. And we know that because we have some subtypes, we have some patients where we actually have the labels and we can benchmark. So that was shocking to us, is the models are actually learning real biology that's clinically meaningful. The other example that's incredibly powerful is once we've trained these models, we can bring in, let's say H&E protein pathology biopsies from a clinical trial where there is a small number of patients, so less than 100 patients, some responders, some non-responders, and we can train a model to predict which patients are responders, and we can do better than, for some of these molecules, we're looking at about half a dozen molecules right now. We're doing better than 75% accuracy at predicting responders from these types of data. So that was incredibly surprising, and we're starting in this case with just H&E. There's no other data modalities. So it's just the beginning, but we're seeing evidence that this approach can work for clinical translation.
And again, this theme of needing to acquire the data, unlike ChatGPT, which could train on all the information in the internet, those kinds of data don't exist. We heard from Tom, we heard from Ron. Now, Marinka, you're focused on this problem of causal biology, and also as powerful as the transformer is, we know that other algorithms are probably going to be even more powerful. Tell us about your work.
Yeah, so a pleasure to be here as well. Marinka, Zitnik, Associate Professor of Biomedical Informatics at Harvard and Moore Institute. Great molecules need great targets. So we are very much focused on developing multi-scale AI models. And by multi-scale, I mean models that can integrate information about the behavior of molecules and how then alterations to those molecules would affect cellular phenotypes. And from cellular phenotypes, tissue context, and all the way to humans. And so the way we typically approach the problem of target identification is to think about not only genetics first approach, but increasingly also phenotype-based approaches that allow us to first create models that can predict patient response to treatment. But then we probe those models, we inspect those models, and through principles of causal learning, we identify what are the mechanisms by which the models make a specific prediction, and we can identify specifically what cell types, what cells, and underlying cell population the model relies on most when making a prediction about a patient response. Why is that useful? That's useful because it allows us to do what I would call reverse translation, to go back from the predictors of response to identifying molecular factors that could explain a response. And now if we equip that with causal learning, we can start disassociating association maps. So we don't just generate heat maps of gene correlations, but increasingly start pruning out those spurious associations to be left with more causal direct effects that could explain how gene-gene reactions, gene-to-cell relationships would really explain the response that we're seeing in patients. And for phenotype-based approach, our rationale for the models that we're developing is that of identifying those specific perturbations. And perturbations can be now sets of genes or individual genes in terms of individual or combinatorial targets that would reverse the disease phenotype and make it shift it closer in this AI latent space towards the healthy phenotype. And so now, under that paradigm, we can now describe phenotypes in a very multi-scale manner. It can be recorded through single-cell or transcriptional information, or increasingly single-cell multi-omics data, or even patient tumor microenvironments readouts if they are available. How do we test this type of AI predictions? So when we get now very long lists of candidate targets, we do two things. So first is we do tremendous amount of prioritization and ranking based on lines of plausibility and multiple evidence that is derived from existing knowledge that is captured by large reasoning models that are trained on not only scientific literature but also experimental data, and help us find those targets that we can be most confident about. And second, we are pairing our computational work with the work in vitro systems, increasingly organoids, that allow us to quickly, in a matter of several months, then test a larger number of predictions, those that are result of high prioritization, to then see whether the phenotypic effects that the target is predicted to restore going from the disease to healthy state is actually observed in that system. And so that allows us then to generate more data that goes into the second round of deprioritization and assessment of targets in a way that is also kind of specific to patient population from which targets were initially derived.
Great, thank you, Marinka. Michelle, tell us about Medable, and tell us about how you're using AI to really transform the whole acceleration of clinical trials.
Sure. So I think everyone in the audience, can really understand from our panelists like this amazing state we're in preclinical science. Also, you can kind of hear that we're blurring the spectrum between preclinical and clinical with some of this advanced work. At Medable, we're very focused on our mission, which is to enable effective therapies to reach patients faster by transforming the clinical development part of the equation, so the in-human testing. So, before founding Medable, I was a resident and a physician at Stanford caring for rare disease patients with a disease called systemic sclerosis. And I was researching this disease at the bench and then also caring for patients and looking at investigational or off-label therapies. And I love the bench science, but I found the in-human part of the testing was really the biggest bottleneck. So founded Medable, as a Silicon Valley startup and have some amazing team members here today. And what we've done is use technology inflection points to solve for this in-human testing, the clinical development part. Our first bottleneck we addressed was at this inflection point of cloud and mobile. And we use that to address access for patients as one of the key bottlenecks. Now I think is arguably the most exciting technology inflection point of my lifetime, which is this cognitive revolution, right? And what's, I think, the most interesting part of this for clinical development is arguably still bottlenecked by the fact it's mostly done by humans. So 50% of clinical trial budgets go to humans. And when you look at what that work is, at least 50% of it is tactical. And that means that, we aren't necessarily applying our brains and our skills in the most impactful way. So at Medable, and I think broadly in the industry, we see an opportunity to use agentic AI to really do this tactical work and to ultimately remove the human bottleneck so that we can get, ideally, a much higher throughput in the system. Because it's like today we have our foot on the hose going from preclinical to clinical. We believe that agentic AI, in doing some of this work with AI, will really transform the speed of development. And I think about my own work as a physician and the ambient AI that everyone, I think, in clinical medicine is so excited about. That's taking the information in the room and scribing it, enabling the doctor to care for the patient, to spend more time with the patient. This is the opportunity of AI. We've seen physicians' job satisfaction go up. I think that we can apply this level of strategic shift of the performance curve for humans across the clinical development spectrum for key jobs that we're seeing a lot of burnout in. And that's ultimately what we're going after at Medable today. So very excited to talk more about it.
And Michelle, you have a slogan, 111. Do you want to tell us what that is?
So our 111 vision is this, kind of crazy idea. It's like colonizing Mars in the clinical development world, where we say, could we get to 1 day patient enrollment, 1 day study startup in 1 year of the development timeline? So, taking 12 years down to 1 year. And what that requires is a fundamental rethink of how we do clinical development. And to your point, Marc, how do we compress the time it currently takes? And so the 1-1-1, we see the biggest lever is AI at this moment in time. And we think we can achieve this, even in the next 5 years in certain clinical examples, certain therapeutic areas.
And just a little bit more granularity, how many clinical trials are you involved in currently and how many geographies?
Sure. So as many people probably know, clinical development is a global process. It's kind of like the only form of universal or global healthcare. So we operate in over 60 countries worldwide. Our technology is deployed into over 120 languages, and we've now been a part of over 250 registrational clinical trials. In phase 2, phase 3, big oncology, weight loss, vaccine. So we've had an opportunity to have a front row seat in what is clinical development and where technology can provide the most promise and opportunity. Thanks, Marc.
Thank you. Reverend Ciucci, tell us a little bit about your mandate and your role in the Vatican, but also now help us step back and think more broadly about AI and the application of AI to human health, and of course specifically this topic of drug discovery and development.
Yes, I'm a Catholic priest. My English is terrible. I'm not a co-founder of Catholic Church. But I live in the Vatican. Yeah, no, I work in the Vatican. Yeah, and in the Vatican we have very few certainty and a lot of questions. Many questions in my mind. Okay, and one is connected to data, models, objects. I know you use Excel. In the Vatican, we use Word. It's a different word. But if you need data, object, and Excel, the question, this is my opinion, the question of a technical approach to human life is, what's human? What's the human? How sounds how this question sounds good for you? What's human? No, I think that it is not enough, it's not correct. What's human or who is a human person? What's or who? The switch is not so insignificant. Two consequences. The first, I think we are more than our data. More. And we are more than the representation you produce to manage and to work. And we are more than what artificial intelligence systems are able to produce and to offer to your scientific research. We are more. How can we preserve and manage this difference? Not only in a very optimistic vision, not to reach your goals. Because I think you are working not to cure a digital twin, but a real person. How to manage the difference. This is one challenge posed by the artificial intelligence today. And the second is, what is a word connected to an object, to a thing? Things, goods, wares. It is very easy to move from a technical approach to an economical approach. How much costs a human life today? And how much costs in New York? How much costs in Latin America? How much costs in Africa? Different prices. I think that what is a good question, but is not enough.
Great. Thank you, Reverend Chu. We'll come back to those themes later today, but everybody, this is a great substrate for thinking more deeply about the work that we do, the impact on access and on thinking of people as individuals and helping them as individuals. I want to, in the interest of time, because we want to have time for some questions, I'm going to ask one more question of each of the speakers, and that is, they told you about their own work, but I'd like now, in your own domain, to broaden a little bit, and for example, in the case of Tom, tell us about molecular design in general. What can we hope and expect? Molecular design, you do small molecules, but there are other modalities. We work on large molecules, and there are others as well. If we look out 5 years, 10 years from now, what should our aspiration be and our expectation for the application of AI? What are the obstacles to getting there? I know data is a big one. We've already talked about this. But what do you see as, again, the promise and what's needed to get to that promise for the field in general for molecular design?
So we don't look at AI in any way as a replacement of human ingenuity or a substitute for that. We look at it really as the right framework for enabling intelligent decision making. There's many heads of research here, many leaders of many sorts of drug programs in the room, and you've all been in project meetings where you are faced with a question of whether or not we're going to go after scaffold A or scaffold B, whether or not we're going to keep making molecules along this line of inquiry versus transitioning to a different possible way or a different modality for going after our problem. And at the same time, you are resting upon, if you're in a big pharma company, decades and decades of all sorts of different sorts of data, which don't immediately relate to that problem, but somehow indirectly inform that set of questions, right? So we look at AI as the right way to take that corpus of data and to translate that into actionable decision-making that might be posed in the context of very intelligent scientific human questions, should we do this or should we do that? Should we prioritize this or not? But then gives you a meaningful and actionable way to say, this is more likely to lead to success. The way that we do that, of course, is through, and I think that the field is increasingly doing that, is through models that can not only make a prediction, but can make a statement of probability about whether or not that prediction is going to satisfy my target product profile. If I keep making molecules in that direction and I make 100 molecules, what is my best guess for the likelihood of hitting my TPP? 10%? 20%? Well, what if I go that direction? Is it going to be a different probability? So I think that in the near term, this is really a very powerful way in the context of molecular design to really be driving that, that thinking. And as we look to longer terms, already those sort of predictions can be automatically turned into synthesis through robotic liquid handling and plate-based biology and all of those things that we and others do. And increasingly, you can imagine abstracting the human questions to higher and higher levels as more of that becomes automated, as more of that becomes incorporating the different dimensions of the question. But that's where I kind of see the field for now in the immediate term.
Maybe I can build on that before we hand it over to Ron and Marinka. Because what you said reminds me of a saying, I think a very profound saying by Eric Schmidt, which is that AI is to biology what math is to physics. AI is to biology what math is to physics. And a good example of that is if you think about aeronautics. You go back 100 years with physics and physics of flow, we had the equations.
Yeah.
We didn't have supercomputers, so it was hard to solve them. So there was a lot of doing some equations, doing some empiricism, going to the wind tunnel, coming back to make a new aerofoil. Then supercomputers came along, and we could actually solve those equations much better. So much more of the work is done in silico, and much less is done in the wind tunnel. And with biology, people tried for 50 years to write down equations for biology and failed. AI is the right tool. So if you feed the right kinds of data to AI, the AI can see patterns that we don't see. That's what it does. Two things you can do with AI. One is for it to see the patterns that we don't see with our eyes. The other is to generate new entities, generative AI, to make movies or to make new molecules. And so the promise of AI at the highest level is if with the right kinds of data, to have a model that understands things, so you can do much more of the work in silico, which is exactly what you're saying, Tom. Assign the probabilities, and less time in the wind tunnel, less time in the wet lab, less time in the clinic, right? So I like that analogy. I think it's actually true. I think it's very profound. There is a problem of explainability, which Marinka told us about. You have an AI model. In what way does it understand what's happening? Can we get below the hood? But now turning to Ron and Marinka, your thoughts about, in general, the problem of deconstructing disease, which is what you're doing, and matching targets to patients so we can make better bets on the right targets and matching them with the right patients.
I like that term, deconstructing disease. I was going to say, fundamentally, in order to make a therapeutic, you need to have a definition of a disease. So, we need to define disease in some way. And so I would say, at a very high level, what we are doing is, trying to identify new definitions of disease that are more relevant for making therapeutics. It turns out that, we've already been using neural networks to define cancers. So, a human looks at a slide and they use their neural network in their brain, and then, they named different features of cancer biology, and we try to sort of correlate those features to treatment outcomes. Turns out that it's pretty hard to do. I mean, we're amazing, pathologists are amazing, humans are amazing, but there's a lot of data that it's really hard for us to integrate so many different data layers. So really what we're trying to do is replace that human neural network with a transformer neural network, and be able to integrate more data. So we're not just looking at let's say the pathology specimen, but we can consider the pathology specimen as well as the genomics of that tumor. Also consider what's happened in the clinic before. If you get to the point where you can start integrating all of this information, then I think you can start to make progress. And so one view of AI just broadly is, historically in science, what we've done is build technologies that allow us to see smaller and smaller things, beginning with the microscope and, all the way to the sequencer. A view of AI is we are not, that's not a technology that helps us see a smaller thing, but now it's technology that helps us integrate more information across many different layers. And so that's our view, is we're going to use AI to integrate all the information we have, get to these new definitions. And today we are, in cancer and generally in disease, we're at a point where we're just trying to name the disease at the very beginning and decide what treatments to use. But you can imagine in 5 years from now, if we can solve that problem, then this becomes a more dynamic process. So we already know that patients that are treated with drugs, tumors evolve, they become resistant, and we really don't have a way to sort of begin to assess changes in response over time. So eventually, as these technologies evolve, as we can use different modalities, hopefully we can make treatment of disease a lot more dynamic.
Marinka?
Yeah, I think for target identification, the current AI models and the current architectures are particularly suitable because of the following property that broadly transformers have. So let's just for a moment look at the word apple. So the meaning of the word apple depends on the meaning of other words within which apple occurs. We can buy an apple, we can grow an apple, and it can mean a fruit or it can mean a company or stock. And so to understand the meaning behind the same sequence of letters, really look at other words within which this word occurs. And so transformers are based on just mathematical, algorithmic definition of and are able to capture this precise effect. And I would argue that it's just so incredibly important for biology because what it allows us to build are these contextually adaptive models. So for example, H2AFX is a gene that is pleiotropic gene. What that means is that the function, the biological pathways it will activate depend on the biological environment within that gene and its products are found. So similarly, as we can look at apple and other words within the word apple occurs, we can look at the gene and its products and other, not words, other molecules, other biological environment within which that gene and product is currently active. And now this is such an important capability that these type of models have that allow us then to make prediction about the role, the function, the activation of pathways for a specific gene or protein. And the models are able to adapt their outputs based on the environment in which they operate. An environment can now be defined by a cell, healthy cell, or a disease cell. It can be defined by an environment where there was a specific candidate compound administered in that biological environment. And so we can start asking much more precise, contextually adaptive questions, where when we ask an AI model, what is biological pathway that this gene will activate? Well, it depends. If it's in T cells, it might be something. But if it's in a different tumor microenvironment, it might be a different biological function. So this gives us just incredible precision to then start looking at, given the definition of the disease that we might get from large-scale analysis of patient data, and seeing what are the pathways that we should alter, modify, target under that definition of disease. Then look at the targets that in that specific context, in that specific biological environment, would most likely kind of reverse those disease effects. So this kind of contextual learning with the ability of generating ultimately virtual simulators that would allow us to look at the effects of different kinds of disease perturbations on the molecular behavior, I think is just a powerful concept for target identification, target assessment that is tied so well with the current developments in algorithmic AI.
And again, this kind of contextual adaptation was just not possible. We didn't have the mathematical tools, we didn't have the compute, and we didn't have the data. The data, of course, we have to generate. But that's why this is the AI moment for that kind of inquiry. Michelle, your view of, again, building on your 1-1-1 vision, your view of accelerating clinical trials in general.
Yeah,
where we're going to go.
So it's, if you look at the history of machine learning, kind of starting in 2010, it was really about prediction, neural networks, deep learning, a variety of machine learning techniques. We get to 2022, 23, and we're at conversational AI, right? The emergence of the large language model. I think, where we are in 2025 is this idea of agentic AI. And this is, what is agentic AI? It's AI that can achieve outcomes and achieve goals. It is AI that can learn along the way. And this, I think, is a really important distinction because as humans, we are outcome-oriented. We can achieve goals. We can be given, a task and know that there's 1,000 steps in between where we are today and how we achieve that task. Something Marinka said that I think is very important when you think about agentic AI, and this isn't just the large language model. This is the surrounding technology to build agentic AI. It's all about context. So if we take a step back and look at clinical development, let's take one job, like a clinical research associate. This is a human who is working across different clinical sites. They're looking at maybe 15 different clinical systems. And today, without AI, they're going into each of those systems saying, how did this site perform in my EDC, my ETMF, my you name it, and actually needing to generate a global view of how that clinical trial is running at that site. They also need to deal with strategic questions in the trial. How is this site going to enroll more patients? What are the strategic things that we can do to actually make this clinical trial move forward faster? Not just the aggregate the data, write the follow-up letters, file all the documents. So when you create technology that provides the context of that tactical work, where you're saying, this is the job to be done, go across those systems, give me a view of how this trial is performing at this site, file those documents, write those emails. AI can do that. Agentic AI can do that work. It's way beyond a large language model, because if you think about a large language model, in and of itself, it's been trained on the internet. Just randomly sampling the internet, you'll find that the data is actually not that great. So what large language models are able to do is provide this kind of cognitive system that understands human language, but you need to ground it and provide the context that's very rich for our world in life sciences. So when I look at clinical development, how we accelerate it, it's about building systems that provide the context for this work and enabling that work to be done by agentic AI so that humans can do the really strategic, hard things that also requires to physically go to places and make sure we understand that physical context. And, for example, on the CRA side, we built a CRA agent. So it's a clinical research associate agent. It works with a CRA. It does about 40% to 50% of that job. But the human then can take that time gained back and actually do the strategic work in the clinical trial. And similar to what we're seeing with physicians and ambient AI, CRA job satisfaction goes up. So I think something that we've been talking about is how you go from the technology to the human impact, not just the economic impact. I believe that the underestimated benefit of AI is it will actually make our lives as humans better, because we'll be able to apply the parts of our brain, the things that matter to us emotionally, the impact drivers. We'll be able to apply more of our energy there, and we're already seeing that with, say, an example like the clinical research associate, who now can spend more time on the things that really move the needle and accelerate clinical development.
Thank you.
And so, how many philosophers, how many sociologists are working with you in these processes? Because I think this is the point. You're right. That's the point. I don't think that STEM science are the future. No. It's a great, very important, of course, more for women, of course, but it's not the future. The future is an holistic vision, an opportunity to put together different approaches, to offer a cultural set in which we can put. But this means we need to work together. How many ethicists in your boards? How many pedagogists in your labs? That's the point. Because we need to be concrete, to be real, this is the point. And I also means that this choice can help us in solving a very real problem. Today, common people is scared by artificial intelligence. Everybody speak about all the problems, risk, we need guardrails, okay, so on now. At the same time, common people use artificial intelligence in a very naive way. And there are a lot of people that write on ChatGPT, excuse me, could you kindly tell me what's the next, what I have to see, visit in New York? And they conclude, thank you very much. Thank you to you. To who? I don't say thank to my refrigerator for cold water. Thank you very much. Very cold, this water. We need to help common people to understand that our machine, really complex, okay, but our machine, we needed to offer a narrative that make us able to manage these incredible opportunities we have, and solve a lot of problems that these technologies are facing. But we need to change a narrative, and we can do putting together different approaches.
Maybe I can make two comments. One about you saying please to the machine. There's a saying about, I'm Canadian. I'm now an American citizen, but I grew up Canadian. There's a saying, how can you spot a Canadian on a crowded street? It's the person who's saying please and thank you to the ATM machine. We're very polite in Canada. We're brought up to say please and thank you to everything, including, I do actually say thank you to ChatGPT. Every time. Right? I regret to say it's what's bred in the bone. But coming back to your point, Reverend Ciucci, for Michelle, you've told us how you discovered that there's increased job satisfaction, but it's an interesting question. Would it be, maybe you're already doing this, but having people in your organization who intentionally ask, we're focused on the clinical trials, et cetera, and we have someone who's going to focus on how do we make sure that we increase the job satisfaction for our employees or all the people in those clinical trials? Is that something that makes sense?
She's sitting right over there, Jenna Daniels, and she does exactly that because, I think, again, the hard part of clinical development is the human part of it, but that's the beauty of it.
Yeah.
So if we're not moving the needle for the patients, and frankly now I think the entire ecosystem, we can't lose sight of that. That's why we're all here. That's this question of what makes us human is the fact we really care about that. We're emotionally wed to one another, to making our lives better. So yeah, I think to your point, Mark, in this whole conversation and about AI, how do we elevate the impact for humans across the board is a core goal. And I think it should be a bigger part of the conversation.
That's great.
At least in Silicon Valley, where you don't, it's more about transformer architecture.
That's right, which is important too. We have some time for questions from the audience. I believe we have some microphones also. So if you can please raise your hand. We have someone over here. Perhaps can we get them a microphone? Okay, someone's coming through. Just so everybody can hear, and also people who are at the monitors outside who couldn't get into the room. Where's he's right there.
Okay, go ahead.
Right at the front row, yep.
I'm just thinking about all the cost of all the thank yous and inference cost.
Yeah.
I've heard please, but I haven't seen thank
Him
you.
first or me first?
Hi there, good morning everyone. Thank you so much for a great conversation and a fantastic panels. I can't help but think that there's an elephant in the room when we think about the potential impact of AI in terms of clinical development in life sciences. It's that, yes, AI is a fantastic engine, but you're saying, each one of you, that it relies on data.
Yes.
AI is a car that needs fuel. We need gas stations. We need data sets at scale. In the context of oncology, the data set it's not just pheno, it's pheno plus omics, and it goes beyond the DNA data at this stage. And the analogy that I have, which for me is problematic and I'm seeking a little bit of reaction, is that, yes, we're seeing the AI engine, the car, becoming better and better, but there are no gas stations. And when you need gas, you need to go drill. And it's very, very large to get the right quantum of clinical genomic data at scale, curated and normalized. And so what is your perspective? Because we're seeing all of these in silico development around digital twins, trying to map biology and deep biology as a system to create models that are predictive in terms of trial success. So it's a little bit going towards efficacy control arms. These models are really not progressing for the past 2-3 years, and they're not going anywhere. And if you add the revenue of these companies, I mean, it's a fraction of a fraction. It's not a rounding error. It's invisible in the context of full, total, global clinical trial budget. So how do you think about the fundamental problem of how do we create those gas stations of data for the AI engine?
And so maybe, you asked about clinical trials. We'll turn to Ron first, but maybe I could just say in the context of molecular design, Tom already emphasized how they have a model, they'll generate data, they'll make molecules, generate data, and feed it back to the model to improve it. In our case, we're focused on large molecule design. We're designing antibodies using AI. And over the past year, we've made about 1 billion in silico designs, and we've tested about 1 million of them in the wet lab, just to give you an idea of the scale which we're generating data. We can get a lot out of improved algorithms and mining information that's already out there, but we recognize we have to generate more data to go to the next level. So that's on molecular design. I think you're right. On the clinical side, actually the problem is even more complex because human biology is more complex than molecular design. So Ron, and you told us about how in the context of immunotherapy, some patients respond, some patients don't respond. You go after very curated data sets that you've generated yourselves and analyzed. Is that part of the gas station?
Yeah.
Yeah, and so we'd like to start there, and maybe Tom would like to add a few words as well.
Yeah, let's see how hot I want to be with this take. So, I think there's one fundamental problem, and so as we're sort of trying to solve some problems in technology, oftentimes you hit a roadblock, and then the solution is actually enabled by something that's step function, many years later. And what happens is, if you're working on the problem in sort of the first phase, oftentimes the solution is sort of nonlinear. It's like you have to do something else in order to solve the problem. So a lot of people have been working on like digital pathology and genomics, and it turns out that the solution to training models on this data is not let's just generate more H&E. Like people are releasing more H&E models. They don't do that much better. The solution is actually to get better, different data. And if you have multimodal paired data where you have pathology, you have transcriptomics, and you have genomics, and you have protein with the same exact cells, the model can see the same cells, you actually don't need 1 million patients. If you want to brute force, like I have 10,000 H&E samples, and I have 10,000 genomic reads from another patient sample, and then I have 10,000 IHCs. If you're trying to sort of integrate all of that into a model, it is a brutally difficult task, and you need probably more data than exists anywhere. But if you can reduce the data set to sort of like a really the right data, just broadly speaking, then all of a sudden you find that you actually don't need as much data as you think. And the problem in biology is we are just never going to be in the regime that we are in with the internet. Like we cannot get the data of the internet. And so you kind of have to reduce the problem to a data set that you can build.
Tom?
Yeah, I really like this question because it's something that we think about a lot. There are certain types of data that can be manufactured at scale, right? You can do 1 billion protein design, or in silico designs, you can make millions, you can run cell lines all day long. You can't industrialize, for ethical and practical and cost reasons that on the clinical side, right? So developing models that can provably get better at, say, clinical endpoints on the basis of scaling, even for different compounds or different programs, stuff you can manufacture at a preclinical level is really an important demonstration. And one thing that we've published in the last year is that in the context of human clinical PK, we can do more preclinical assays, in vitro and early in vivo assays, on different molecules and make a prediction for human clinical PK that beats the state-of-the-art models with 20-fold less clinical training data. So, and when you include all the available training data, it sets a new standard. So it's really, I think, something that these transformer models hold promise for, to take seemingly disparate data that you can manufacture at scale and point that at questions that are intrinsically unscalable in terms of the data creation.
I think a lot of people are finding that if you have been able to train a model on data that's easier to come by, not necessarily easy, but easier to come by where you can generate lots of it, and then you fine-tune with data that's harder to come by, there's this phenomenon of transfer learning where the model will provide insights into the smaller data set that you couldn't get just by analyzing that data set alone. And that's what's behind a lot of what's being called the virtual cell. You might have heard about this, another announcement yesterday by the Chan Zuckerberg initiative, of generating lots of perturbation data to create a model, a causal model of cells There you have to do it with cells that you can interrogate at scale in the lab. And they aren't necessarily the cells that you really care about in the human, in the disease state. But that provides a scaffold then that you can fine-tune with harder to come by data to get insights. At least that's the theory, and I think that will play out. Maybe Marinka, would you like to comment on that? No? And Marinka will have the, actually no, Father Ciucci will have the last word. But,
Last word.
No,
Last word. Last apocalyptic. The last word is excellence. You are excellent in your research, in your economical results, in the scientific results. But please remember, if you can offer an excellent product only for a few, that's not good. We need, I hope you can reflect on what does it mean to be excellent, real excellent. I think we need to introduce the word justice in our excellencies. Because if we offer a product, an excellent product, only for a few, you are normal, not eligible for Galien prize.
I know there are probably way more questions. I see someone even has the microphone. I think we started late, and in the interest of time, we'll have to get off. But the speakers are here, and I think we'd be happy to continue the conversation offline. So thank you very much, everybody, and thank you again to the panelists.
Thank you very much.
So interesting.